usually very embarrassing because every time a famous scientist comes to this meeting, and we have many of them, people that are well-schooled, the first person they always want to speak to is Steve Fowkes, okay? And Steve I asked him a question here a couple of years ago. I'm standing there and I said, Steve, I printed this off from the Genome Foundation, you know, the thing that they've spent hundreds of millions of dollars back east on. And I said, is this paragraph, what does this paragraph down here mean? And so I handed it to him. He's looking at the paragraph, and at the same time, he's crossing out the chemical formula that they have up there and changing all of the chemical formula on the sheet. And I said, Steve, what the hell are you doing? He says, these fools, they're spending hundreds of millions and they don't even understand this very simple chemical formula here. And so I don't know whether he was pulling a scam on me or not, but all I know is that Steve over the years has been terribly, terribly helpful to everybody in this organization. When I encounter a very serious problem down in Monterey, for people that I know, the first person I want to get on the phone is Steve. And Steve will get on, and he'll spend an hour or 2 hours with the people on the phone, and it's just absolutely tremendous. And I notice that everybody that comes into town from anywhere in the country, the man they always want to speak to is Steve Fowkes. So he has a great, great deal to say. And I would strongly recommend, Steve, if you'd be kind enough the next time you come to our meetings to bring some more of what you call your bricks. Now, these are all of Steve's things. A number of you people have them in this room. I think they're about $75 from all of his websites and all of the medical letters that he published over the years. Now, I think almost everybody in this room is taking Depronel as a result of Steve continuing to pound on that. And I always like to use the story because I think it's very important. And that is that rats that will live to be, that are physiologically identical to us, I actually recognize it in some of the people that we have here. But they live to be 145, 155, 165 weeks old. They then lose their fur, they lose their intelligence, they lose their mobility, they lose their sexual ability, and then they die at a maximum of 145, 155, 165 weeks. If they're given Depronel, and it's all covered in the book that Steve was the co-author of, Smart Drugs 2, and he covers it in one of his things, and just that alone is worth the price of that $75 for all of his publications. It's absolutely amazing literally what happens. So many of us take a couple of drops of 1 or 2 or 3 drops of Depronel under our tongue, and literally what it does, the rats, when they gave it to the rats, within a few months they grew back their fur, they were more mobile, they were more intelligent than they'd ever been, their mobility was strong, and I think their lifespan went to 225 plus weeks. And so I think it's vitally important to understand that, just simply with the thing that you were talking about as far as collagen is concerned. I have everybody, all of my tennis playing friends down there, okay, and they're ending up beating me because they were more mobile to begin with. These guys are 40 years younger than I am, and I insist that they take collagen. I insist that they take vitamin C, ascorbic acid. And it's absolutely amazing what these things do. So everybody in this room, everybody reads, everybody is smart, everybody is intelligent. And my old teachers, they'd be surprised to hear me thrown into that category. But basically, I think we owe so much. We owe so much to this man right here. And so, Steve, let me bring you up here from one great scientist to another.
Testing.
Jim, do you want Steve
Loud
to
enough?
use this or not?
Can you turn it down?
You sound fine.
Is it okay?
Is that okay? Good.
Okay, very good, Steve.
My new toy. Turn it on first, right? Okay. I'm going to start with the end of this presentation so that those of you who want to get up and leave immediately can do so. Okay, I'm going to talk about ways to prevent and reverse Alzheimer's disease and senility. And it's not going to be definitive because how about that set? No, I mean the ones in the back would be. What do you think? You want the extra? Okay. Can you guys read that? In the back?
One side off is better.
Okay. There's a lot of chemistry, a lot of biology, a lot of physiology involved in dementias. And given the fundamental complexities of the brain and the support systems of the body that support the brain, there's dozens of things that can go wrong. And any one of which can result in dementia, and several factors of which are involved in Alzheimer's disease. So all I'm going to do tonight is paint the picture of what we know. Even though the Alzheimer's organizations and doctors won't tell you this information, the science is there. We now know enough to do something about it. And my focus won't be to tell you which doctors to go to. It'll be how to tell you how to do it yourself at home. For yourself, if you want to prevent cognitive diseases, or for other people to work on them once you have them. So the rule is, for prevention, you can do it yourself. Earlier is better, but you can do it yourself because your brain's still intact. But for reversal, you can't do that. Earlier is critical because damage can accumulate. Okay? So if part of your brain dies, it's gone. If it just goes unconscious, you can wake up and have full function. If you get hit in the head with a football and you're lying on the ground unconscious, you wake up and you may have 99.9% of your functionality back. But if you have Alzheimer's for a year, it may not be that effective. So what is dementia? Well, there's a whole series of definitions of it that circle around certain issues of thought, mind, ability to reason, as distinguished from emotional or motor control functions. But you can have, in the first definition, emotional disturbance that's secondary to the cognitive dysfunction, or they say cognitive decline that is more than you'd expect from normal aging. But nobody really knows what that is anyway, right? But there are people who live into their hundreds and who are fairly bright, and other people who in their 50s start to decline, and so there's a huge variation that we see in this. And so I hope this talk will inspire you to take some steps that I'll present later to monitor and intervene to protect your own brain. I think we're now living in the age of the brain, and that most of us earn our living by brain instead of brawn. Okay, so all of these different kinds of dementias, this is just the tip of the iceberg, but major categories are here. So what do you see here? What's the theme that you can see in terms of these? Okay, I'm going to give you a brief hint here. Okay, what's above the line and below the line? Okay, there's a mechanism involved below the line, supposedly a cause, some kind of underlying factor. Above the line, it's defined by a context. It has nothing to do with any hint that tells you what you can do about it. It's like, you know, old age. What are you going to do about that? But if you look at the below the line, you know, we still don't, on some level, know what's going on. What is it about dialysis that causes dementia? It's not like if you don't have a kidney that you can really avoid dialysis. But it would be nice to know, is there something you can take to prevent you from getting dementia if you do have to put up with dialysis? Traumas, you can't go back and not have an automobile accident. pugilism, you know, you can't go back and not be in a fight that you were in. Syphilis, should hit you with the laser beam, right? Alcohol, dementia. What's involved in that? I mean, we know that alcohol consumption depletes B1 right here, but the symptoms of alcohol dementia and B1 dementia, they're not the same, they're different. So what is it that's going on in alcohol that's different from a B1 deficiency in terms of producing dementia? So basically we can drop this line down and say, well, only a tiny fraction of these do we have an actual cause where we really know what kind of underlying mechanism is involved. And so this is the real problem that we have. How do you deal with something where you have no idea what's going on? So what do we know about dementia? Well, obviously it's the brain on some level. So we can talk about what's unique with the brain. We have metabolism that's unique, phosphorylation reactions, for example, that are dominant reactions in the brain that are not dominant anywhere else in the body. The cellular morphology. Brain cells are long and drawn out. An average dendrite in a brain is like a 2-lane highway running from San Diego to Maine. That's unique morphology. How can you expect a normal cell to operate under that kind of difference of structure? In terms of function, neurons have electrical potentials on their surfaces that other cells don't have. And the brain is isolated behind the blood-brain barrier. So there are things that the brain will be protected from because of its unique sensitivity. And so we can say, you know, talk about alkaloid poisoning. So certain kinds of mushrooms you might eat, chemicals found in plants, the blood-brain barrier rejects them. A nutrient, choline, your blood-brain barrier rejects that. It prevents your brain from becoming overstimulated from choline. Certain kinds of infections, blood-brain barrier protects against the invasion of the brain. Insulin resistance. Insulin resistance lowers the energy that cells can derive from glucose if they're GLUT4 receptors, but not if they're GLUT1 receptors, and the brain is located, has GLUT1 receptors. So the insulin resistance affects your body more than it affects your brain. So you have people who have diabetes who are actually mentally quite sharp. The brain is more sensitive to other kinds of influences. I mentioned, you know, for example, getting hit on the head with a football. You get hit on the arm with a football, you end up with a bruise. You know, you get hit on the head, you go unconscious. So your consciousness goes to zero in a very short period of time with a relatively minor stress. But carbon monoxide poisoning, lead poisoning, hypometabolism, and that goes to the issue of energy. So it turns out that the energy that the brain operates on is a critical factor involved in dementias. The brain is roughly 3% of your body and consumes 20% of your energy. So that is a special factor that turns out to be critical in Alzheimer's disease and almost always critical in dementias. So this is why we have dementias in robust people. Otherwise, extremely healthy people lose their minds. This is also why people with severe body diseases like diabetes are mentally sharp. And it's also why certain areas of the brain can degenerate and not other areas of the brain. Parkinson's disease affects the motor control centers, but your cognitive function can be literally unimpaired. So, energy. How many have heard of the second law of thermodynamics? Cool. Basically, this is the tendency of the universe to degenerate into chaos. That disorder happens in the world. And biology is an example of an anti-entropic process. We create structure. We defend ourselves against this chaos. And when we lose the battle, we die. And so entropy is a concept that actually describes the fundamental challenge of biology, and that is we have to get energy out of our external environment so that the entropy in our bodies is opposed at the expense of increased entropy out in the world. So it's a trade-off. Because we're an open system, we're not violating the second law of thermodynamics. All it means is that we're living at the expense of our environment. We're getting our energy from the environment. Oxygen from the air, glucose and fat from our food, nutrients, and our body is putting this all together to maintain its and defend its structure. And the brain has massive structure issues. I mean, trillions of neurons and trillions of synapse connections. It's a very complicated system. ATP, adenosine triphosphate, mentioned earlier, is our most common form of energy. It's the chemical currency of the cell, and it fights entropy. And GTP, instead of ATP, GTP is an alternative form of energy. It turns out that it's critically used in certain brain structures. CTP, creatine phosphate, is a backup energy supply. I'll show you about that in a minute. So a lot of this function about the brain is about conserving and generating ATP to keep your brain systems running. But beyond that is something called reducing power. And a lot of people in here have heard about this. This is what vitamin C is promoted for. This is what NADH is promoted for. It turns out that the environment that we live in is extremely oxidized, massive amounts of oxygen, which is poisonous to the living process in us. And so we have to be very careful to absorb that oxygen and use it properly in order to generate the energy. And our body structures themselves are very reduced. So our bodies are rich in electrons, which means reduced, and the atmosphere is extremely deficient in electrons, and it's this potential between the outside and inside that we use to generate the energy to live. Okay, so it's this energy that gives us the ability to be advanced life forms. So we had to be aerobic to evolve from a single cell into multi-celled, and we have to be aerobic in order to develop brains. This 20% of the body's energy is a massive amount of overhead. This is to make everybody pass out. But I handed out sheets to people. I hope you got one that you can refer to this, but I'm going to be pulling this diagram up interspaced throughout slides throughout the whole thing because it'll allow you to focus on the overall theme of the mechanisms involved in Alzheimer's disease, and the rotating red circle will tell you the area of the diagram I'm talking about at that particular point in time. So I'm going to start with the energy production systems of the body, which is basically the mitochondria. This is where sugar is burned for energy and where fat is burned for energy. This is where ATP is generated and oxygen is consumed and CO2 is generated. So here we have CO2 is generated from this system. This can have a negative effect on cognitive function. We have ATP and GTP, which can have a negative effect. Positive effects flow over this way into the antioxidant defense systems. So I'm going to tie all these things together for you in one big picture, and then you can figure out what to do with it afterwards. Okay, so here's a diagram of the difference between what gives us the ability to have brains and other organisms the ability not, or the inability to have brains. And so basically this is the single cell dominant life forms of bacteria and other single cell organisms. Eventually they became aerobic as single cell, and then they piled up into multi-cell organisms. And what is different here is the fuel. Glucose is the fuel for the anaerobic system, and glucose or fatty acids can be the system for the aerobics. They both generate acid, but lactic acid is the byproduct of anaerobic metabolism. How many of you have had aching muscles from overexercising? That's lactic acid. How many heard Schallenberger talk about his aerobic training systems? Yes. And this is exactly what happens when you overexert and you go beyond your capabilities. You generate lactic acid, which comes out of the cell and makes your muscles ache. But in the aerobic system, you get CO2 as your primary waste acid. Okay? So what do you get from 2 ATP? You get life, single cell life. What do you get from 38 ATP? You get multicellular life and ultimately organisms with brains. This is a diagram of the mitochondrial system. I'm going to go through it really fast. All I'm going to do is show you two parts of it. NADH, complex 1, which is right there, complex 1. And this accepts the electron from the NADH, which is in blue, indicating it's cold. And then we have at this other end, the electron passes down the chain to complex 4, where oxygen, which is red and hot, is the acceptor of the electron. So the NADH is shoving the electrons into the system, and the oxygen is pulling them from the other end. This is what drives the energy production systems, and we end up with protons pumping through these complexes, which then drive a generator to give us our ATP. So the NADH is a critical transfer for making ATP. NADH is an antioxidant. It's a reducing agent. It powers the antioxidant defense system, but it also generates ATP. So whether you're talking about energy on one side or reducing power on the other, they're kind of the same thing. And so the antioxidant function of NADH down here is what defends your glutathione and vitamin C, and I'll show how that ties into dementias in a little bit. Okay, here's your backup system. So you're producing ATP. Well, you have a few seconds of ATP available in your system. Well, how does your body handle the need for maybe needing more ATP? Well, there's a compound called creatine, which then gets phosphorylated. So the ATP here donates the phosphate to the creatine, and now you've got the phosphate in a storage form. And this sits there until you run out of ATP. And then it runs backwards, converting back into ATP. So this is like a backup power system or a generator where you're pumping water up into a tower, and then when you need the power, it comes back down again. Turns out this is very critical for cognitive function. So muscle cells, nerve cells. Well, in nerve cells, we know this is powerfully inhibited by mercury exposure, which is a causal factor in Alzheimer's disease. 95% inhibited in Alzheimer's disease. Basically, it's wiped out. Memory problems, cognitive impairment, lack of strength, lack of stamina, all of these are manifestations of the inhibition of creatine phosphate. Let's move to blood flow next. Okay, in order to have energy, you've got to deliver the oxygen, right? You've got to carry the CO2 away. That's hemodynamics. This is, I'll apologize, this is a very complicated slide, but there's some really important information here. How many people have heard of congestive heart failure? Cool. It's a very serious disease where the heart runs out of energy like the brain does in dementia. And so ketones, which is what happens when you burn fat, when your liver exports ketones or your cells generate ketones locally, these increase the heart ejection fraction in mice, in rats, with congestive heart failure in just 30 minutes. It's a phenomenal change. And according to most medical people, this kind of change cannot happen spontaneously. But I'm telling you that ketosis can generate it spontaneously. Also, that if you're, well, let me skip that one. I got to go through these in about 2 minutes per slide, so pardon me if I go over things fast. Blood hypercoagulation. Okay, this is caused by inflammation, and I can testify about this personally. Amen! I had this problem about 7-8 years ago. I had 2 simultaneous fungal infections, my blood got hypercoagulated, and I could not remember what happened 5 minutes ago. I could read a paragraph 5 times and not remember what happened in the beginning of the paragraph. That's how bad it was. Well, I took nattokinase, and 2 days later, I could think totally normally again. Okay. Heparin can do the same thing. There's also several other enzymes that can potentially do the same kind of transformation. So if somebody has dementia, maybe it could be because their blood is coagulated. And if you give them nattokinase, their dementia will ease and change. It's a simple thing you do. Go down to the health food store, buy some nattokinase, give them 1 capsule the first day, and then 2 the second day, 3 and 4, and see, does their dementia go away? If you're trying to prevent dementia, let's say you play bridge, you do video games, you do crossword puzzles or something, and so you do your own nattokinase challenge and assess, are you thinking better? Are you performing better? Is your bridge scores competitions higher in the duplicate tournaments? Or do you beat your video game scores? So this can be practically applied fairly easily with something as simple as a video game. I'll come back to those kinds of tests, self-care tests, at the end. Lung disease, gas exchange, emphysema. My grandfather died of Alzheimer's disease when I was in college, which is why I'm here talking to you about this stuff. He had emphysema. So his lung function limited the oxygen delivery to the cells. pH disturbances in the blood can alter the binding and release of oxygen and CO2. Goes to the concept of metabolic balancing or acid and alkaline body types. You can test this out by, for example, breathing CO2, changing your breath, rebreathing, power breathing. Breathe in for 4 seconds, hold your breath for 4 seconds, exhale through pursed lips. For 16 seconds, you drive the CO2 back into your lungs. Does that affect your cognitive function? Does it affect your memory? Does it affect your strength or stamina? If you do the opposite, hyperventilate, overbreathe, do you get dizzy? Do you pass out? Do you develop memory problems? So you can load up on the CO2, you can deplete the CO2. Find out how it works for you. Plaque in the vascular system. Goes to the issue of vascular dementias. Lysine C and proline, Rath's formula, can be very useful for reversing that kind of condition, strengthening the collagen and making the vascular system less sticky. Magnesium, vitamin D, high-dose vitamin D therapy, we've heard presentations about that. That can reverse soft tissue calcification. So there are ways of going after these kinds of conditions that you can then assess through cognitive testing. Is it working? Magnesium deficiency, again. Nitric oxide deficiency. You can do a challenge with nitric oxide. Does it improve your cognitive function? If you've got somebody with Alzheimer's disease or dementias, does it improve their cognitive function to some degree? These are things that are easy to test nowadays. You can't find it with your doctor, but you can do it on your own. You can buy arginine, ginkgo, and vinpocetine from your local health food store and take all 3 of them and see if that changes your blood flow and your mental function. Okay, fuels. We got glucose and ketones here and here. They're feeding through the bloodstream into the cells to generate energy. Glucose can become impaired. This is from a slide I helped prepare for a paper that was a lead article in Nutrition Reviews on ketones, and it was called Ketosis, Metabolism's Ugly Duckling. And that's because there's been a huge prejudice against ketosis in the medical profession because they grew up, back at the turn of the century with diabetics dying from ketoacidosis. So they automatically assumed that burning fat was pathological. Turns out it's not at all. So what you can see here is that we have the yellow pathways are your ketone pathways, beta hydroxybutyrate, alpha beta ketobutyrate are the fuels. They flow straight in. There's no resistance effects, no transport problems. They just flow right into the cell passively. On the other hand, here's glucose. We have glucose delivery problems, we have glucose transport problems into the cell, we have the receptors, interference in receptors, all of these different factors here that are sabotaged by known pathologies that interfere with glucose utilization for cellular energy. So relying upon glucose is highly problematic. And in the United States, it's even more so. Insulin resistance is endemic. Overeating, right? Ketone fuels. This is going to be the real meat and bones of this presentation because this is, in my opinion, a core intervention for maintaining your cognitive function and aging gracefully. So basically, when you're in ketosis and you're burning fat, you have 3 potential things that can enter your system. Hydroxybutyrate, ketobutyrate, which is also called acetoacetate, which is the archaic name, and acetone. Well, this is if you're healthy and your ketosis is working properly, it's 80% hydroxybutyrate, 20% ketobutyrate. If it's not that ratio, you can have problems because you end up with too much ketobutyrate, or you can end up with a generation of acetone, which gives you acetone on the breath. This happens when people go into ketosis to an extreme degree, and like fasting, for example, can happen, and they are inefficient about being in ketosis. And so this is one of my cautions to clients, is when you go into ketosis for the first time, be gentle. Do it carefully. Do it deliberately. Give your body the chance to burn ketones, to learn how to burn ketones. Some people in this room may not have been in ketosis for 40 years. Many Americans may not have been in ketosis since they were teenagers. When you have unlimited amounts of food to eat, you do not go into ketosis, okay? Or at least unlimited carbohydrate. So, how do you know? Well, there's something called keto sticks. You can buy them in any drugstore. And you touch the tip of the strip to the stream of urine, and a little bit of urine is absorbed onto it, and it changes color if you have ketones. Goes from yellow to pink, brown, or in between red, and that is reacting to the ketobutyrate or to the acetone. It's not reacting to the hydroxybutyrate, which is 80% of what's being generated. So fundamentally, your testing system is hinky. That's a very scientific term. Acetone is not a fuel. You can consider it to be a low-grade poison, and the liver does not metabolize it, so you have to outgas it through your lungs to get rid of it. So if you're going to go into ketosis and you're going to go in strongly, you may want to have somebody around to smell your breath and tell you whether they can smell acetone, and if they smell acetone, back off. Consume some carbohydrate. Ketones are preferred over glucose by most tissues by a factor of 10. If you put a cell in a cell culture and give it a choice, ketones. Ketones are probably favored, this is a viewpoint of mine that I've observed from the literature, they're favored with development and aging. In other words, the ability to burn fat is part of the process of developing. It's part of the process of maturing. At each stage of development, you notch down your glucose metabolism and notch up your fat-burning metabolism in age 1, age 2, age 7, at puberty, and this continues slowly throughout the rest of our lives. And so forcing our bodies to burn carbohydrate by overeating carbohydrate is pushing us the wrong direction from our normal developmental process. And this is why carbo consumption is associated with cancer, and why I think that one of the first things that anybody with cancer should do is cultivate ketosis. And contrary to scientific myth, the brain is not glucose dependent. 50% of your energy can be supplied. If you're metabolically efficient for burning ketones, 50% of your energy can be supplied by ketosis in 2 to 3 weeks. Okay, so how do you get into ketosis? It's pretty simple. It's your backup energy supply, so you create a brownout. You stop eating carbohydrates, and your backup system kicks in gear. Very simple. You may not think that if you've got severe carbo cravings at that point in time. It can be quite stressful. But most people, when they're efficiently burning ketones, their carbohydrate cravings go away. So it is a temporary thing. You just need willpower for 3 days or maybe a week. 2 different diets. The Atkins diet, which most people are familiar with, which works fairly well in young teenagers and young adults. And then the Ezrin diet, Calvin Ezrin, who was one of the first proponents of the use of ketosis for therapy for insulin resistance. And his is high-fat, moderate protein, which reverses that. And in my opinion, the fat is fundamentally safer than the protein for middle-aged and older people. You can facilitate your transition into ketosis by fuel. Short and medium chain triglycerides don't need carnitine. They just slide right into the cells and get burned. So you start burning fat instantly when you consume MCTs. So butter is 5% MCT. Coconut oil is roughly half. Coconut and palm oils are roughly half. And a synthetic oil, MCT oil, is 100% MCT. There's no long-chain fat at all, which means none of it can be stored. It's the long-chain fat that we have in our fat deposits. And those require carnitine, which I'll explain later. My butter recipe, which I really like, it makes it easy to change butter. If you mix butter 50-50 with MCT oil, it has the same profile as coconut oil. You can facilitate your fat burning by taking mitochondrial nutrients. I think I'll just go on.
Steve, are you saying, in other words, butter is actually helpful in order to get yourself into ketosis?
Well, it's better than olive oil, which is 0% MCTs. But 5% isn't that great. Yeah, I mean, it's a relatively small amount, whereas a tablespoon of coconut oil, gives you a teaspoon and a half of medium chain triglycerides, which means you have an immediate effect on your energy. And when you're in carbo withdrawal, in the initial stages of trying to induce ketosis, you have less energy than you normally have. And so if you have Parkinson's disease, your symptoms are worse for a while. Your tremor may be worse. If you have, one of the factors I'll go into a little bit is carbo cravings related to serotonin deficiency. Well, there's depression. So if you have depression, that can get worse until you get into ketosis. So a lot of what I'm going to be talking about is about, being in ketosis where your ketosis metabolism is efficient. But if you haven't been in ketosis in 10 or 20 or 40 years, that may not be the case. There are inefficiencies in the system. It doesn't get tuned up. It's like buying a used car that's been sitting on the road. You got to take it out on the freeway and burn out the gunk. And that can be painful in a sense in terms of the experiencing those toxins coming out of your system. A lot of things like baked goods in the United States use partially hydrogenated vegetable oils, which are inhibitors of ketosis. And so, if those are in your fat deposits, you're going to have to clean those out before you're going to be a really efficient fat burner. And so, part of this is about cultivating it. So you go into ketosis, you go out of ketosis, go in and out. Each time you do this, you get more and more efficient. You clean out more of the gunk. Your transition becomes smoother and smoother until you can do it without any stress at all, with very little effort. And so as you tune up your systems, less of your ketobutyrate and hydroxybutyrate spill into your urine. Your cells are soaking it up like dry sponges instead of wet sponges, and so you see the color change shift from the darker colors into the lighter colors, and that's a sign that you're becoming efficient and you're doing exactly what you want to do. So the color strips change not only because you're not measuring the most plentiful form, but because over time the efficiency of the system is changing underneath the detection system. These are supposed to be livers and hearts. This is the actual distribution for being in ketosis, where your body is burning most of its energy in terms of fat. It's the liver. On my screen, I can actually see it's bright red, but up here it's so faint. But the liver takes your fat, your adipose tissue, and converts it, breaks it down, and generates the acetoacetate and beta-hydroxybutyrate, which are your ketone fuels, which travel through your bloodstream to your muscle, your heart, your brain, and then the process reverses to generate ATP energy. So the liver is taking the fat and chopping it up into little pieces. Your bloodstream is delivering it to you, to your cells, and then they're burning it for energy. That's the kind of specialization of function that the liver does very, very well. And just to show you how well this has been worked out, this is the diagram from that article that shows all the metabolism that's taking place in the liver regarding the export of ketone fuels. And so you can see there's lots of reactions taking place down in this area, which is the inner compartment of the mitochondria, and then the fuels flow up out into the blood where they're distributed. There's all kinds of complicated systems where things can be tuned up. Carnitine, which has been mentioned earlier, which I will mention again. Carnitine is the transporter here that brings the fuel and transports it through into the cell to be burned. So long chain fats, long chain fats and very long chain fats require the carnitine for transport. Medium chain and short chain do not require carnitine. It just flows right into the cell. So that's why coconut oil is a really, really good clean fat to incorporate in your life if you want to cultivate fat burning. Okay, antioxidants. And a special interaction is the glutathione portion of the antioxidant defense system. How many people here have heard of glutathione? Okay, good. Glutathione is the primary cellular antioxidant. This is what defends the cells against the oxygen in the atmosphere and protects the cell membranes, protects the cell structures in the center of the cell. And there's an antagonism between glutathione and mercury. So glutathione basically does two things. It defends the cells as an antioxidant, and it detoxifies mercury. And so I'll talk about these two dynamics as they relate to Alzheimer's disease and dementias. So NADH is your reducing power. This is driving the system. Converts to NADPH, which is your primary nucleotide reducing agent, and that defends against defends the glutathione. So when glutathione oxidizes, interacts with a free radical and oxidizes, then the reducing system's converted back into glutathione, so it can be recycled again and again and again. So NADH is your recycling for your antioxidant system, and it recycles glutathione and ascorbate. How many of you know that the explorers in the Arctic developed scurvy when they ran out of food? Yeah, it was bad. And when they started eating blubber, what happened to their scurvy? It went away. There is no vitamin C in blubber. So what is it about blubber that caused their vitamin C to regenerate instead of oxidize? It's fat burning. It's ketosis, which is exactly what I'm talking to you about. So glutathione contains an SH group. Chemically, that's a sulfur and with hydrogen. This is the active reducing portion of it. And this is sets the redox potential for all living systems, is this sulfhydryl group. And it does mercury detoxification. And the primary ore in the earth is mercury sulfide, which is cinnabar. And so mercury and sulfur have a special affinity, and in biology, this is how our biological systems defend against mercury, is we bind it with sulfhydryl groups, tie it up, prevent it from poisoning other things by giving it a sacrificial material, which is glutathione. So whenever you have a problem in your body where mercury gets loose, all of the enzymes that depend upon these sulfhydryl groups get poisoned. This shows you that mercury is a risk factor in Alzheimer's disease. This was a study that was done not by the American Dental Association. But the toxicity of mercury turns out not to be the key factor. It's not just an issue of exposure. It's an issue of the strength or weakness of the mercury detoxification system. So there are people with high mercury levels who don't have cognitive problems, and people with low mercury that do, because their mercury detoxification systems are either robust or weak. Okay, now we're going to go down into the kinases. And these are enzymes that are unique to the brain. Well, there are kinases outside of the brain, don't get me wrong, but in the brain they're incredibly active, there's huge numbers of them, and they're constantly in action. And so that part of it is unique. And the kinases and phosphatases do opposite things. Okay, so kinases are enzymes, they add phosphate groups to enzymes, and the phosphatases remove them. The kinases also operate on kinases and phosphatases. So there are some kinases that when they get phosphorylated, they increase in activity, and others, when they get phosphorylated, they decrease in activity, which ends up producing a cycle. Every 90 seconds, the brain oscillates from going to an overphosphorylated extreme back to an underphosphorylated extreme every 90 seconds. This is one of the reasons that your brain uses 20% of the energy of your body, is because it's doing this. It's doing these whole series of reactions and then undoing them. That means that a lot of energy is being wasted. But it serves an entropy function. How many electrical engineers and computer whizzes do we have in the group? Okay, anybody ever create an oscillating circuit to create long-term stability? It Turns out that's a much safer way to create stability than a damp circuit, which tends to drift over time. Well, if you create an oscillating circuit, you tend to maintain stability over a much longer period of time, and so that's what this is doing in the brain. Protein kinase C, massively expressed in the brain over any tissue, and it's 50 to 90% inhibited in Alzheimer's disease. Okay, microtubules. This goes to the question of the brain's morphology. These long, thin dendrites, this two-lane highway running from San Diego to Maine. How are you going to move food from San Diego to Maine, or lobster from Maine to San Diego? You're going to put it on a truck and move it there. Well, what happens if the road disappears? You're going to put it on horses. You're going to have people carrying it with their sneakers. It's going to take, instead of 3 days to get across the country, it's going to take 3 months to get across the country. This is what happens in Alzheimer's disease. This is a diagram of a microtubule. It's circular. Proteins are arranged in an external shell, and they polymerize length to length from these tubules to create this tube, and this is like a road. There are little motors that are ATP driven that either move one way down the train, down the road. This is a polarized road. It has an end out at the periphery, at the end of the cell, and it starts at the nucleus of the cell, so it's directional. So there are motors that will only carry things outward bound, and other motors that will only carry things inward bound. And these things are very efficient. They move things over very long distances. This is how a nerve cell lives. This is how a nerve cell survives. It's how a nerve cell functions. If the nerve cell cannot move material down these roads, it stops functioning. Doesn't mean it dies. So if you have Alzheimer's disease in an early stage, all these roads are gone. All you have to do is restore the roads, and your brain starts functioning again. So it's only when these dendrites and axons in the nerves die and necrose and disappear that you have permanent damage in Alzheimer's disease. This subunit here is inhibited by mercury. It's inhibited in Alzheimer's disease. There's actually a model where the scientists have actually demonstrated the addition of mercury to a cell culture is the only agent. Aluminum doesn't do it. It's the only agent that depolarizes the growth cone in neurons. And there's actually a snail neuron, a marine snail neuron, its neuron is big enough they can actually take a movie of it. So they actually have a...
They're aggravated by zinc.
I didn't put the slide in about that, but I can go into that if I have time at the end. If you'd like to raise that as a question, I have a slide stuck at the end that I can show you about this. Turns out zinc and mercury are isoelectronic, which means they have the same outer shells. They're right below each other in the periodic table, along with cadmium. And so zinc and cadmium and mercury share a kind of toxicity, and they also share binding to sulfur. So zinc binds to glutathione, and cadmium binds to glutathione, and mercury binds to glutathione. And so there's people who believe that you can aggravate Alzheimer's disease with zinc supplementation.
What about root canal?
Root canal teeth are sites for potential anaerobic bacterial infections. The normal surveillance of a tooth comes up through the nerve channel, and the white blood cells come up in and they go out the tubules in the tooth, and when they run into a bacteria, they eat it. And so there's an immune surveillance of the entire interior of the tooth, which is hollow. But when you do a root canal, you seal off the center pathway so nothing can get in there, and the bacteria that are in there just grow. The outside of the tooth is permeable, so nutrients can get in there, but there's no immune surveillance at all. So once the tooth becomes infected, then that's an anaerobic infection, and there are chemicals that are released by those bugs that poison the brain and act like mercury. I also have a slide, if we have time at the end, you can ask me about that. I can show you a compound called gliotoxin that does the same damage that mercury does, and it binds to these sites and enzymes, and it's irreversible binding. It's a one-way thing, and it's produced by root canal teeth. Yeah. was the ApoE relationship between the maternal and the paternal ApoE, and you have the 2, 3, and 4 versions, and so you have these 9 potential combinations, and it turns out that if you have the 2, 4 genes, which are the high risk, you have the earliest onset, the next one is the next earliest onset, and so it goes down the line, basically 10 years between each of these, and this particular group here is so rare that nobody's ever really observed that with any degree of statistics to provide a number, but it's probably, people beyond 100 years of age before they get Alzheimer's disease. So these are the risks, and the 45-degree angle across the diagram. And so here's the difference. What is the difference between ApoE2, 3, and 4? The 2 protein has 2 cysteines, and the ApoE4 has 2 arginines, and the natural one is in the middle. And so the correlation is that cysteines are correlated with lower risk, and lack of cysteines is correlated with higher risk.
Steve,
Yes?
stupid question. If there's 4 variants, which I thought there were, where's the first one?
There's 3 variants. ApoE2, 3, and 4.
Oh, okay, well, there's 3 variants.
There's probably was an ApoE1 that they came up with in rats or something, but once it gets named, it never gets unnamed.
Okay, yeah.
So there is no ApoE1, at least, that is common enough to worry about it. And so here you have your 2 diagrams, and so you see an exact correlation between the risk and the cysteine content of these enzymes. And so the hypothesis is that since we know that sulfur binds, the cysteines are sulfur and they bind to mercury, that somehow this protein, that nobody really knows all of what it does, is somehow involved in carrying mercury out of the brain. Okay, so here's the inflammation mystery. So we know mercury is involved in Alzheimer's disease. Fish contains high mercury, so why does fish lower your risk? Omega-3s. Yeah. Anti-inflammatory fatty acids, fish oil. EPA and DHA produce anti-inflammatory prostaglandins, and it turns out that inflammation is involved in diseases, both neurological and non-neurological, and so inflammation is a bigger risk factor than mercury by far.
So could humans would help?
Yeah. All kinds of things directed at that issue of inflammation are potentially quite valuable, and some of the research is showing that. Mm-hmm.
What does the inflammation do? I mean, we know what the mercury does. It breaks down the microtubules. What is the inflammation?
Well, that's a good question, and I'm going to talk a little bit about this, but I certainly don't think I know the whole picture, and I'm not going to be presenting a definitive picture of what's involved in inflammation. But certainly, inflammation inhibits healing. It inhibits you get certain cytokines and neurotransmitter alterations, and so these have potential effects on energy systems, which I'll outline. But if anybody else comes up with something else, please let me know.
There's also, I gave a lecture on cancer and inflammation. There's also a link of MMP, the
Metalloproteinases?
Yeah,
Uh-huh.
they're shown to be a major factor in cancer, but also in Alzheimer's.
And are elevated or lowered levels?
Elevated.
Elevated.
And so they're working on MMP therapies to, as part of the inflammation, you have to have inflammation in order to have MMP.
So do they know anything about whether or not the MMP overexpression causes distortion in mercury, zinc dynamics, or copper utilization, chelation?
It does more damage to the neurons. It causes increase, almost like fibrosis. It's almost like a new collagen. It's like, when you cut yourself, you have MMP, and you have new collagen developing.
Well, fibrosis is an alternative to collagen.
So that
Okay.
causes disturbance in the blood.
Okay. Well, let me, yes.
Excuse me, on the MMPs, actually they become quite a factor with Lyme disease, and so I've looked into it quite a bit for that, and have been taking Huzang, which is also polydom caspididum, to control that and keep it down, and actually in my recent testing, my inflammation levels have greatly reduced since I started taking that. So.
Mm-hmm. Yeah. Yeah, so this whole issue of the immune system cascade and the cytokines, the cell signaling factors that are activated by inflammation, is extremely complex. I'm not going to go into all of it, but I'm just going to lay out a couple of things here for your edification. So inflammation induces cytokines. Cytokines are hormones, cell signaling factors that are released by the immune cells to signal each other to do things and coordinate the activity and defenses of the body. And two of these compounds that the cytokines systems interact with are aromatase and IDO. IDO stands for indolamine dioxygenase, and tryptophan is an indole, an indolamine. So the aromatase increases estrogens. So it converts testosterone into estradiol and androstenedione into estrone, and I'll show you a diagram of that in a second that outline how that works. But estrogens inhibit your energy systems. So it's like turning down your voltage. And this is good when you have a fever. Okay? You don't want your energy systems adding to the damage that's being caused by the immune system, going to have to fry all these foreign invaders and have your own cells damaged in the process. So when your immune system goes up, you want all your other cells to duck and run. And that's what estrogen does. And you get massive production of estrogen in a fairly short period of time during a fever. And this is also what happens in older men. Why older men will have higher estrogens than their wives is because after menopause, estrogen goes down in women, but in men it goes up for their entire lifespans. And this is activated by infection. So this dominance of estrogen plays a role in energy.
May I please ask a question? You mentioned a moment ago that cytokines induce aromatase and
IDO.
and that is related to tryptophan?
Yes, and that's right here. Okay, so the IDO catabolizes tryptophan. And the reason it does this is because tryptophan is a growth factor for microbes. Bacteria use tryptophan to grow. If you've got high tryptophan in your bloodstream, the bacteria are going to grow and your immune system is going to have a bigger challenge. So it drops the tryptophan down to keep the bacterial replication down, assuming that the inflammation is caused by bacteria, and then your immune system is supposed to mop up the damage, and then the inflammation goes away and your tryptophan levels come back up again.
So would it be better not to be taking tryptophan if you have inflammation issues?
Sure.
Okay.
I mean, this is one of the things that I've noted is people with inflammation problems, when they take tryptophan, they don't get the sleep benefits that they do when they're not inflamed. And this I noticed myself. I've developed a trick to get around that, but it's a real problem. So
5-HTP converting to it?
5-HTP is also destroyed by IDO. Tryptophan and 5-HTP, which are the only precursors for serotonin, they're both destroyed by the IDO induction. Okay, so now your tryptophan is going down, so your serotonin goes down and your melatonin goes down. Okay, these have significant effects on your endocrine system. Your decreased serotonin increases your sensitivity to pain, fibromyalgia, sensitivity to pain, depression goes up, and sleep quality goes down. Sleep quality gives you sleep disturbances give you neuroendocrine destabilization. They cause your rhythms to be disturbed. They cause the healing process that takes place at night to be aborted. Well, that healing process may play a role in the brain in terms of potential emerging dementias and Alzheimer's disease. Okay, what happens to decreased energy? Steroids go down. Okay, we mentioned earlier about the production of estrogens here. Well, estrogen is just one of the steroids, so you have increased estrogen and decreased steroids. So you have less protection from progesterone and pregnenolone, which are your two primary neuroprotective agents. Let me come back to that in a second. Okay, so also because testosterone and progesterone upregulate your energy pathways and the estrogen turns them off, you're not only flipping the off switch, you're sabotaging the on switch. So here's the steroid tree. And so what you can see here is that the cholesterol starts here, converts to pregnenolone and goes up, DHEA, into the testosterone side. We can say this is the neuroprotective sector of the tree, which is basically your trunk. And the progesterone and pregnenolone, so this end of that space is the most neuroprotective, and it gets weaker and weaker until it goes over to here. And you can actually grow human nerves with DHEA in a culture, but it's very difficult to do. Okay, next we have the androgenic sector up here, and these are the strong androgens, testosterone and dihydrotestosterone, and then go down to the weak ones, DHEA. And then we have over here, we have the estrogenic influence in this quadrant here. And then over here we have our neurotoxic one. I'm sorry about the blue not showing up in this light. But the point is that you have the effect of aromatase is these arrows right here. And so what they're doing is they're moving stuff from the androgenic into the estrogenic sector and from the neuroprotective up into the estrogenic sectors. And this is bad news for the neurological system. Okay, causes of inflammation. I'm just going to give you a few highlights here of things that are very obvious. There's a lot more to it than this. Viruses, certainly, bacteria, protozoans, fungi, parasites, spirochetes. All of these things are organisms that we can get infected with.
Steve, on the last slide, so things like Hemodynamics would be helpful.
Yes. So if the inflammation pathway is active, then the enzyme here, that pathway right there, that's aromatase, that arrow. And there's an arrow here, that arrow is active, that's aromatase. And this arrow there, that's aromatase. So taking an aromatase inhibitor blocks the conversion and compensates for the over-estrogenification from inflammation. So if you have chronic inflammation, you can turn it down by taking an aromatase inhibitor. Mm-hmm.
So are you defining inflammation as the presence or absence of certain chemicals?
No, well, no.
Or molecules?
There are molecules that are associated with it and agents that are associated with it, but it's basically a process in the body, a metabolic shift in the body. It is a defensive mechanism. It's why it's there, but it's designed to deal with infections. And it's designed to deal with things like sunburn, as a stress, an acute stress, or a burn, or a wound. And so it's not supposed to be on all the time. But when it does get turned on all the time, because you have a chronic infection, your body doesn't clear it, or you have food allergy, which I'll talk about in a second, where what you're eating is triggering the immune reaction that's taking place, it becomes chronic. And this leads to a lot of downstream consequences, like fibromyalgia, autoimmune diseases, and I don't know, somebody want to chip in here with some contributions?
Then what about a root canal? Is that something then you want to find the right person to do it? Do you need a root canal?
Oh yeah, if you're going to have a root canal, it better be done by somebody who understands not to use gutta percha as the filling agent for the center of the tooth, and who knows to sterilize the tooth to whatever degree possible before it gets filled. After it's been ground out and you're supposedly down to something that is solid, to use like hydrogen peroxide or chlorine dioxide in the tooth to sterilize it.
Sterilize the tooth.
To try. Ozone, yes. Ozone is very good for killing biological systems. Fries the polyunsaturated fatty acids, and that can kill things. So ozone is very good, and it also, because it's a gas, penetrates down the tubules into the far reaches of the tooth very well.
Dr. Kim in our group specializes in biofilm.
Mm-hmm. Yeah. So you can say herpes, and the reason I put herpes here is because I don't know if any of you have heard the stories. There have been several stories in the news in the last several weeks and going back for months. The whole viruses cause Alzheimer's disease hypothesis has now been resurrected by a group of researchers who did some, I don't know if I can even describe it as idiotic experiment, but this has been going on for many, many years where people are looking at associations and mistaking the direction of the causal arrow. So if you have A associated with B, it doesn't mean A causes B. B could cause A, or Z could cause both A and B. And so they don't get this kind of complexity, and they jump to a conclusion, And so what they're really trying to do is sell us a lot of Zovirax. So anybody with Alzheimer's disease also should be taking Zovirax, and so they get a big, huge captive market for their drug. So again, I put root canal teeth in here as a potential source of infection. Nanobacteria being something that's very rare and controversial. Toenail fungus being something that is extremely common. It was one of my 2 fungal infections that caused my dementia about 7, 8 years ago. Chronic allergies is another source of inflammation. And this can be the immediate allergies, hypersensitivity like rashes or pollen allergies, where you get a reaction in your symptoms in minutes, sometimes seconds, but certainly minutes and maybe hours. Very quick reacting. And these are IgE-mediated responses. Ig stands for immunoglobulin, which is part of your immune system, antibodies. And then there are delayed antibodies from food allergies where the reaction can be spread out over 8 days. And so if you've got food allergies, chances are you don't know it, because there is no immediate experience for most people unless you do a very, very careful cleansing and reintroduction. And even then, it may not be clear.
There's an excellent blood test available, though, that tests about 200 or 300 things, and you can get a pretty good idea of what you're allergic to.
Which one is that?
I just bought it actually from ImmuPro, and I'm like allergic delayed in 22 things, including things like nutmeg and mustard and eggs and chicken and dairy. And
Do you know if it's cytotoxic testing?
They came up with the answers were IgG
Okay.
answers.
Well, cytotoxic testing, though, is not necessarily a definitive measure of the immune response. And so it may be valid in different people to different degrees. So I wouldn't necessarily trust it.
It's blood. It's serum.
Yeah, but cytotoxic testing uses the cells, but it's measuring a particular kind of reaction. It's not definitive for the entire possible range of pathologies that could result from this kind of problem. So if you test positive, that means something is going on, but if you test negative, you're not really sure that something might be going on that you're just not measuring. Yeah. So in terms of this is a huge area that could potentially be studied by people looking to prevent Alzheimer's disease or senility syndromes. Leaky gut syndrome is a common breakdown in the healing process. If you have high estrogen levels for a long period of time and constant inflammation, your gut healing may be compromised. There's a lot of activity that takes place in the gut. The immune system is incredibly active there. There's a lot of toxicity from chemicals that come through the gut. And so if the gut gets leaky, then you have undigested food particles that pass in that cause inflammation. And so this is, wheat allergies and Crohn's disease and these kinds of things are manifestations of that kind of problem. And the bad news is that common foods, the foods you eat every day, are the ones most likely to produce these allergies, and your favorite food is the one at highest risk. Oxidative stresses are sources for inflammation. Heavy metals, nutritional deficiencies like selenium, which is protective, that can play a big role. You have chemical exposures, aldehydes like formaldehyde and from particle board and carpeting to acetaldehyde from drinking alcohol. These are all chemical insults to the system that can produce inflammation. So alcoholism would be an example of a lifestyle factor that can do this. Almost there. Okay, so I've got 3 summary slides. First, I'm going to talk about dementia. So the brain is uniquely sensitive, and so if you pay attention to your brain, it can tell you what's going on. It can tell you whether you're likely to develop dementias, whether you're on the road. It can tell you whether anything you do increases your risk, decreases your risk, and it can tell you in somebody who has dementia whether or not what you're doing is making it worse or making it better. So any one of these systems I've been talking about can be the key factor in a particular case of dementia, or it can be some combination of a whole bunch of them. So this is not a simple problem. Each system must be investigated to determine whether it's involved. We don't have blood tests to test this stuff. So it comes down to trial and error. So you can do medical tests like trace mineral profiles, heavy metal toxicities. You can look at poisons in your fat. You can look at antibody reactions to your white blood cells. All kinds of tests that you can look at that can kind of like be a shotgun to kind of point the direction that maybe you should be paying attention to this factor before you pay attention to some other factor. But at-home self-care testing is the key, in my opinion. This is what I train my clients to do. You test yourself, you test your cognitive function, you let your brain become part of a biofeedback loop to prevent or reverse dementia. Alzheimer's disease summary: 3% of the body mass, 20% of the energy. So this goes to maintain the microtubule transport system, cyclic phosphorylation, dephosphorylation, and a host of other reactions that are just more active in the brain than anywhere else. These require the aerobic energy systems because you get 38 ATPs instead of 2. They're exquisitely sensitive to mercury. Glutathione binds to mercury, and glutathione recycling is the critical process to Alzheimer's disease. Okay, this is a brief summary. I expect you can give me some questions about this. To find out how your brain is working, you can go to a neuropsychologist. Some of the doctors in the area will have people like that on call, and they have all these tests that they've worked out. How good are you at recalling a 5-digit number backwards? If you get a story, somebody tells you a story, can you remember something that happened in the first part of the story? These kinds of tests. You can be trained by these people to do them at home or do them with a relative. The primary limitation with these tests is that they're done relatively infrequently. So you don't get a lot of data points. So it's very hard sometimes to make decisions about what's actually going on a minute-to-minute, day-to-day, hour-to-hour basis. So I suggest that at-home testing is the way to go, and the first category is computer tests. Computer games, computer challenges, because the computer system is really objective and it tends to do things the same way every time. So you come back and you're tired, you come back and you're fresh, and the computer doesn't care. The cascade failure design gives you your best results because when you reach a point when you make your first error, that automatically increases the difficulty of the test and causes more errors to happen, and so it all tends to come apart all at once. And so whether it happens here or here or here, it's really a good indicator of how well your brain is doing. Even so, subtle differences in brain function manifest as significant differences in your test results. And Tetris is my favorite one of that category, but there's a game called Typo Attack that teaches people to touch type, and so that's another example of that kind of thing. Letters drop from the sky and you have to hit the letter on the keyboard to make it disappear. It's kind of like the game with the rays shooting the alien spaceships. But any cascade failure type of design will work. And so, in terms of picking them, my suggestion is you pick the one that you like the most. It's the most fun to play. If you're going to do this every day, it ought to not be a drag. Concentration, a card game you play with a stopwatch. How long does it take you to clear the table of cards, matching them up 2 at a time in pairs, and taking them off the table? I played this when I was a kid. Normally I played it with somebody else, and so it's a challenge. You do 2 and they do 2, and so you're learning from them. But you can play it against yourself. And if 52 cards ends up taking a half an hour, you can just do hearts and spades or just do face cards. Reduce the size of the deck to make the test convenient enough that you don't mind doing it on a daily basis. Proprioception tests. Okay, what do they do when you're drunk? Touch your nose. Or do something like this, where you're out of position and touch your nose. So how well do you know where your hands and feet are? That's part of some very complex neurological functions. And it's like balance functions, which is a very good thing for something like Parkinson's disease or any kind of neurodegenerative condition, where you stand on a platform and it measures the swaying of your body. How good are you at correcting for balance problems? And then you close your eyes. And now you no longer have your eyesight helping your inner ear. You're now totally relying upon your inner ear for your balance. How does that then change the pattern of the swaying that you're doing? And then you tilt your head back. Now your inner ear is out of its orientation in unfamiliar territory. So now the pattern changes again. And so you get to see how, is there a kind of dysfunction there, and does it change from week to week to week? You can have a doctor do this, or if you have somebody who can get you a forced sway platform, you could potentially do this at home. But those tend to be pretty expensive and hard to find, so my suggestion is do something that's accessible, like a card game. These are just examples of things that you might do in your daily life, that you could say, okay, I'm already doing this. Let me use it for cognitive assessment purposes. And then you do correlations between these tests and your non-cognitive tests from doctors or whatever, and whatever interventions you're doing, to see if I go into ketosis, does my mental function improve? And this is how you make decisions about preventing things, is getting more and more and more into a groove where your brain is doing as best as it can. Okay, time for questions.
How would something like high fructose corn syrup interact with the system? It starts with your liver and it's going to have some effect on this whole process.
Indeed. I'd say there's a couple of things that it would do. One is it's a sugar, and it's a fast-acting sugar, so it's going to induce a maximum insulin spike, and it's going to suppress your ketosis, your fat burning. You get a massive dose of sugar where the sugar is able to meet all your energy needs. There's no need to burn fat, shuts the fat burning system down.
So we have one guy here that tells us that high fructose corn syrup is an unnatural product in reality, and the liver does not know how to handle it very well.
Well, that's true, but the liver doesn't know how to handle fructose in very high percentages. And so one of the things that happens with fructose is you can get a induced copper deficiency that can happen in your cells. And this is so serious that if you consume half your calories from fructose, a teenager can die in a matter of 2 weeks. I mean, it's potentially that serious. But there is fructose that's found in natural fruit sugars, and so when you eat a piece of fruit, there is some fructose in it. But if you talk about a natural source of having 100 different carbohydrates combined together in a fruit versus only 2, you're now in a realm of having something be dangerous because it's now lopsided. The balance that's there and the flexibility of the system to take a 4-carbon sugar and a 7-carbon sugar and combine them together to get a 5 and a 6-carbon sugars, that's no longer there because you only have 6-carbon sugars or 5 and 7 carbons. That you don't have that flexibility. Or the system in the liver that handles something is overloaded. So you can eat an entire head of light? Good, yeah. I mean, this is a potential problem with any of the sugars if you consume them to excess. Even something like xylitol, if you were eating a pound of xylitol a day, this could be a problem that wouldn't show up if you're only eating an ounce a day. So I would just suggest that this isn't something that, that one shouldn't depart from a natural theme. It's just a matter of wisdom that one would say you want to trust the way that it's been done over ways that have never been tested before, even though you don't necessarily have any data. Just that prospect. Okay.
Yeah, do you prefer NAC over glutathione as this agent for binding mercury? And does
what was the first part of that?
N-acetylcysteine over straight glutathione?
I don't recommend it. The only glutathione or glutathione precursor that I think might be safe is the liposomal glutathione. And the reason for this is that the ability of the NAC or cysteine or glutathione to bind mercury means it can carry it into the cells, that as you ingest it, it serves as a mercury transport device going in the wrong direction.
Would MSM help against that? Because it's got sulfur in it?
No. The sulfur that's oxidized doesn't bind to mercury. And so sulfur monoxide, which would be DMSO, doesn't bind to mercury, and sulfur dioxide, which is MSM, doesn't bind to mercury. So it has to be reduced sulfur to bind to mercury. And so the idea is that with the liposomal glutathione, it's inside a sphere of fat, which means it's protected from the mercury as it goes in, there could be some interaction with mercury, but most of it would then get deeper and deeper into the body before it then breaks open and releasing the glutathione, which would then theoretically carry the mercury out instead of delivering it in. And there's another factor, and that is that there's the methionine receptor at the cellular level, which methionine has a sulfur but it has a methyl group on it, that when you take cysteine and you put a mercury methyl group on the end of it, it looks like methionine. So it becomes, transportable through the delivery on a facilitated basis. In other words, the cell is sucking this stuff up. The poison is deliberately sucking it up because it's mistaking it for methionine. So I would not recommend glutathione precursors. And the other reason that I would caution against them is that the data show that short-term therapy with glutathione precursors raise glutathione levels, but that the regulatory system corrects and brings it back down to baseline. And therefore, you cannot, by feeding precursors, provide sustainable increase in glutathione. And therefore, the focus should be on the recycling of glutathione rather than an attempt to override the feedback mechanism that controls the level of glutathione. Does that make sense? Okay, over here.
Does aloe vera have any type of precursors to glutathione?
No. But it is a mucopolysaccharide that produces potential immune system reactions and could be a source of both inflammation and anti-inflammation. Bern, do you have any experience with that?
On the aloe?
Yeah.
Yeah, you said it right on it. It's more of an inflammatory, it works excellent on inflammation. And the mucopolysaccharides.
Yes.
How do you encourage the recycling of glutathione?
Ketosis. Anything that increases cellular NADH. NADH is a carrying system that's designed to unload the hydrogen, the reducing equivalent, very rapidly. So there's NAD, which goes to NADH, but most of it's in the NAD form. And so, it's like your body produces NADH, and immediately the NADH goes and does some work. But there's something called NADPH, where it's the other way around. NADPH stores almost all of the NADPH is in the NADPH form, and the NADP is a minority component. And so that's what's happening to the NADH, is it's building up that NADPH pool, and that is also building up your vitamin C pool and your glutathione pool, and these are what are defending your cells against oxidative insult, radiation at the dentist, getting on a plane at 7,000 feet, weekend skiing at 10,000 feet, whatever it is where you're exposed to something that's deleterious. If your antioxidant defense system is robust, you're capable of withstanding a greater insult without it overwhelming your systems and causing cumulative damage. Yes. You had a question?
I'll talk to you after.
Okay.
Is use of low-level laser any help in preventing Alzheimer?
The what laser?
The low-level laser application.
Low-level laser? Oh, infrared laser?
Infrared light, yeah?
Yeah, infrared light affects some aspect of mitochondrial function. And I don't know what it is. I don't know, does anybody know what it is? But there appears to be a phenomenon called dark adaptation that takes place at night, which is why the winter is especially hard on people when you have, 12 hours of darkness versus 6 hours of darkness. And that exposure to infrared light in the morning seems to set that up, which is why in my bathroom I have I don't use the lasers, I use just incandescent, lights using tungsten to just shine on my body when I'm in the bathroom so that I'm getting the red end of the spectrum. So it's mimicking the sun when it rises in the morning, when all of the blue light gets scattered all over the sky, then you look at the sun, it's bright red. That means you're getting more of that red and infrared part of the spectrum.
Well, there is lots of information on laser recently, but in Europe it has been used for quite a while.
Mm-hmm. Yeah, I don't know about specifically the role in Alzheimer's disease, but there are several neurological diseases that are of known mitochondrial pathologies, things like alpha-ketoglutarate dehydrogenase deficiencies and things like that with Parkinson's disease. And so anything that could potentially intervene at the mitochondrial level can be powerful, and some of those lasers can penetrate up to 10 inches in terms of going through tissue, and so you can actually irradiate the spine from the front with that kind of power.
It helps in healing wounds.
Mm-hmm.
It definitely, it's proven on that.
Yeah. Yeah. Yes.
Steve, can you make your slideshow available to us?
Sure. Yeah. If you just send me an email, any one of those addresses, I'll be glad to send it to you. Are we going to shut down, or we got room for more questions? Uh-huh.
Are you still doing health counseling?
Sure.
Okay. Now, Steve is one of the great resources. He doesn't promote his business. In fact, he's in the nanotechnology business right now. But he's one of the greatest resources you'll ever find. And I've been present in many of his counseling sessions, and they're just wonderful. So I just want you to know that he is available. He is too busy to bill you, so if you do use him, please remember to pay him, because he's in a startup company, and when you're in a startup company, you're not making
okay.
of money yet.
That number right there.
Right. So he's got a very good system where if you get counseling, he'll set up the tape recorder, because obviously you're going to hear lots and lots of stuff in a fairly short time.
So how many people feel overwhelmed from this presentation?
Right.
Yes. So that's what the tape recorder does. It gives you a chance to listen to it as many times as it takes, and to transcribe it onto paper in the process of
consultation. I found when I use it for various people is much clearer. A lot of this we don't have chemistry backgrounds, and then you're very concise and clear, and you lay it out in simple terms.
Yeah. I try to know my audience.
Let me just tell you a story. I don't know if I've ever mentioned it. About this time last year, I went to Europe to visit my son in Switzerland. We had to climb up the hill to see the old town of Geneva. I couldn't make it. I was very embarrassed because I consider myself dead and so forth. And I couldn't do it. I had to ask him several times, would you please wait? I can't make it to the restaurant without stopping for you. Well, it turned out when I took my pulse, because I thought my heart must be racing, I was working so hard, it was 37. And I couldn't get it up over 37.
Thyroid.
I went to Kaiser, they gave me $5,000 a check, bless their heart. They weren't stingy, they gave it everything they got. I did all kinds of research on the web, I couldn't figure it out. I was talking to Steve about the nanotechnology stuff, because I'm in a small way, a supporter of that company. And I said, by the way, Steve, I got this Palo. I take my heart rate over 37, Kaiser doesn't know what to do. They said maybe I should do an ablation of the nerve or something. He says, Bill, do a thyroid challenge. It's probably thyroid. Well, as it happens, I had cold hands and feet before I sold from Alan Brower. I had some Thyrolar in my refrigerator, which I hadn't used for 2 years. So I went and got one. I took it. 4 hours later, my heart rate came up to normal, 62. It started to be variable again. And I've been taking a half of the Thyrolar ever since, and no problem. So, 30 seconds of consultation solved the problem that Kaiser couldn't solve and that I couldn't solve. And Steve somehow intuitively knew what the most likely problem was. So, how often
Classic.
But I'm not too surprised, because, I guess you guys probably realize this is something rare. You could go to medical schools all over the country and not hear a talk this good. So
I'm going to take questions. If people need to leave, please, it's not rude to leave. I'll continue to take questions, and if we break up, I'll talk to people one-on-one. Yes?
I have just one question. Sometimes ago we had Dr. Bonney who showed that mercury could be removed from the body by magnetic field.
But if you call that number, you'll get me. On me?
Yeah.
I do have stuff, yeah.
Yeah. And where would I call?
You can go to ceri.com and read some stuff about that I've done. CERI.com.
Oh, yeah. Oh, yeah. No.
Right there. Ceri.com. www.ceri.com.
Okay. Good.
Easy. 4 digits.
Thanks.
Yeah. 4 numbers. Okay.
Steve, we had here Dr. Bonney. He removed mercury out of the body by magnetic field.
I've never done that, and I don't know the physics, and I don't know the efficacy. I haven't seen the data on it, but I'm really intrigued by any way of getting the mercury out. But there's a real problem. The number one most sensitive organ in the body to mercury is your brain. The number two one is your kidney. And that's how most mercury chelation goes out, is through your kidney. So it has to be done slowly and smoothly over weeks and months to be done safely. Okay, fecal excretion is theoretically unlimited, and there's no problem in terms of dumping as much mercury as you want. But the mercury that's secreted from your bile and your liver into your gut for secretion gets converted by microbes back into methylmercury, which is then reabsorbed. And so the mercury cycles goes to the liver and goes back through the gut and then into the brain and then back to the liver and into the gut and back to the brain. Getting rid of it is a problem.
So, but you say, do it slowly.
Do it slowly, and it's important, but the issue of inflammation is more important than mercury. So if all you do is take somebody with Alzheimer's disease and remove the mercury, there's no guarantee it's going to work. You know, you have to work on their energy system. You have to get their inflammation down in order to make it all balance out.
Thanks.
You're welcome.
BHT, Steve, what is this BHT book?
I can email it to you if you email me. It's a book on the
Mary and I consistently for a 10-year time period. It was about 12 or 15 years ago. For 10 solid years, we ate that way. From 45 to 55 years of age, somewhere in that range.
I thought you said in 1845 to 19.
And I would go to my cardiologist, and he would do the blood tests. And when I was 45, 47 years old, he says, you have your tests are like a 21-year-old person. And then somewhere in time, I decided both Mary and I simultaneously went off for what we were eating. And then we kept going off and going off and going off. And as we were going off, things started to happen because the chemistry shifted. You can see it. But we didn't know what to do. So what we were doing is that we said, well, we need this, we need this, we need this. But nothing really shifted. And so a couple of people here suggested, first started with Mike, suggested that we go down to the Optimum Health Institute down in San Diego. So for New Year's, Mary and I, instead of celebrating New Year's like most people celebrate, what we did is decided to go have a really mind, body, spirit, clean out our bodies for New Year's. That's what we did. And we ran into so many incredible people that have had catastrophic diseases. I mean, we're talking major stuff. Who have healed themselves, who have gone through the medical profession to the extreme, only to find out that they had to change their lifestyle. And when they changed their lifestyle, they changed their body. And the stories I could tell you for hours because I talk to these people. It's just like unbelievable. So that's the gift I wanted to give. Is it easy to do? I mean, Mary and I had to go through the discipline of 1 week of doing this. And after that week, it was like, I actually enjoy, for the first time in a long time, sitting down, and it sounds kind of weird. When I went to Whole Foods today, you know, you walk around the supermarket, there's only one place I gravitate toward. Guess where that is? Yeah, the color of the vegetables. I said, Mary, before I leave Whole Foods tonight, I just kind of walk on this side. And it sounds kind of weird. Maybe it is. But I think intuitively we know. I think everybody knows. So I wanted to share that with you. We'll see how this goes. Soon I'm going to do my cholesterol tests and everything else because my cholesterol started to go too high. And so now we're going to see where that's going to bring me. And if everything goes back to normal, I think this is going to be a process for us for the rest of our life. Will we deviate periodically? Yes. But when I do deviate from something, it really feels miserable. It only tastes good for like a minute or so. And then my stomach says, what the hell did you do? My body says, this is weird. This is strange stuff. So that's the gift I want to share with people today. So thank you.
Thank you.
Thank you. Right. Mary, what do you think?
He's right on.
All right. I think he's too. I mean, a lot of people have had this experience. I can't remember people in Cameroon who eat this plant. It's a fruit like a mango. And somehow they discovered that they use the seeds of the plant. They can squash them up and use that to thicken their soup. And they have soup as a staple in their diet along with whatever else they're having. And it was noted that these people were very lean and never had diabetes. So somebody said, what's going on here? I mean, you know, they have a problem of weight in Africa, I think, is just like everywhere else. These people didn't have it. So then this one fellow started doing research, and they came up with this this product that if you take this extract of these seeds, which is really a food product in a way, you can overcome leptin resistance. So yeah, I don't want to go into it because we just don't have time, but the article is very compelling. And William Falloon, who's the kind of like the CEO of Life Extension, himself wanted to be sure that this worked, so he tried it, and he said, in a letter that I got, he said, because he said they sent it out to people who bought it. I bought some, gave it to my some of my friends and so forth. But he said, I'm a skinny guy with a pot belly. That's just the way I've been built. He said, I took this stuff and I lost like 4 inches off my waist in 8 weeks. And he said, I still have some to go, but he says it's working. That's why I thought, in addition to the research, that this was on the up and up. So I think it might be a real breakthrough. Richard.
That's if you look up the bellyfatcure.com, he has some videos on there of several people similar to that.
Yeah.
After a very short time, they're losing several inches in pounds. But he also gives certain, Clemmies ice cream. You could eat that, has no sugar.
What kind of ice cream?
Clemmies. I don't know where you get it. And, that soda water that Teddy was drinking. He said only good thing is xylitol and stevia, just like we've been saying. So there's a lot of interesting things in there.
Yeah, okay. Well, there's more than one way to do it, I'm sure.
Yeah, there's somebody in there on the video that shows you what she does to make breakfast.
Right. I mean, there's the juicing way. There's the zone diet way. There's all these different ways, as Steve says, to get into ketosis. When you're in ketosis, then you're burning ketones, which are the fat molecules or complexes that that are used for fuel. Yes, sir. Go ahead.
To change the subject, second opinion, he talks about MMS, and it might even be better than Artemisia, I guess, for cancer and so forth. So,
what is MMS?
It's a
Miracle Mineral Supplement.
It's chlorine dioxide, so it's chloride.
Chloride.
It's a chloride form. It's a chlorine molecule. It gets rid of malaria and viruses, but for cancer, there's a lot better things out there than chloride.
Okay.
It is a good thing for malaria and viruses and bacteria. But you're going to have a reaction, a major reaction. I used it 30 years ago, and boy, it was pretty. The reaction was you have to be very careful because the Herxheim is really bad.
Yeah. Yes. Steve says it's super bleach, so I mean.
Yeah, that's what it is. It's a bleaching item. It's like you might as well do hydrogen peroxide.
I'll tell you what. What I'd like to do now, I gave Mike Clark a little warning on this. I hope it'll work for him. He has a wonderful set of programs lined up for 2009. So I thought we'd have a little preview of 2009. What's coming up? And Mike, are you up for that right now? Good. Excellent. By the way, I want to thank Mike. I got a present for him. Mike has done a great job. He's just an incredible, energetic, talented guy. He not only does he line up a terrific program, but he also works on the newsletter, pretty much as our main editor. So I got him this book. I just found out he didn't have it today, so I got him a copy. This book is fantastic. And I hope, I started to be always using superlatives, but I only talk about the great stuff in here because there's limited time, right? So we only talk about the great stuff. This book here was, again, they had one chapter excerpted in Life Extension magazine, and then I got the book. This is, I think, the definitive book on testosterone replacement. And the research is tremendous, and what a great read it is. I wish I could tell you some of the things that are in it.
What's the name of the book?
Testosterone for Life by Morgenthaler. Morgenthaler. H-A-L-E-R. It's like Morgan Fowler without the H. This is pretty funny.
This is not John, this is Abraham.
Yeah.
Morgan Taller.
Morgan Taller.
He spoke at the A4M last December for those that went. The lineup for this year is next month. We all know what we're having tonight. We're having a Dr. William Clint, who was here 6 years ago, and he presented on brain spec SPEC imaging. Now, those of you who've heard on PBS, there's a Daniel Amen that's been on many times, and he also spoke at the conference on what they can do with that kind of imaging to determine what's going on with you. So Dr. Clint will be here next month, and he's in San Jose and was trained by Daniel Amen. In March, we're having there's a group called the International Stem Cell Institute, that I didn't even know existed, that Aubrey de Grey and others belong to, and they have conferences every year, and things are going to change with the new administration, and the head of that institute, and it's in Florida, is coming out at his own expense in March to speak on what's going on in stem cells. In April, we have, let me skip that one for a minute. In May, we have Dave Steenblock, who spoke here a few years ago on using stem cells for treatment, and he also uses oxygen chambers, and he gave a great talk here a few years ago, and he's spoken all over, and he happened to mention when he spoke here, this was the best audience he's ever come across, including his international ones. In June, Len Saputo in Walnut Creek, I think, is going to speak on his newest developments. He hasn't spoken here in about 4 years. He was waiting for something new, and he's going to announce it then. In July, we have Jim Wilson on adrenal glands. And in October, we just signed up. Did you hear about that, Phil? In October, we signed up that guy.
Gary Taub.
Gary Taub, who's written, I haven't had a chance to read it. What's the name of his book?
Good Calories, Bad Calories.
Okay, and this is something that somebody knows about here. Just mention it briefly.
Okay.
I'm not sure.
You know, I kind of have to apologize for all the superlatives, but in my opinion, having studied this stuff for 13 years, this is the best book on diet that's ever been written. And the reason it is because it encompasses all the other books that have ever been written. And it is a brilliant analysis, and it goes against the conventional wisdom. It shows what a poverty of research actually was behind some of the things that were considered the conventional wisdom. Gary Taub is coming out to speak with us, and also the Commonwealth Club and some other places. And October 15th, and this guy wrote for the New York Times and is really changing everything in the world of nutritional diet research.
And he's also coming at his own expense.
Yeah.
So, by the way, there are some people here from the FMBR group, remember, Foundation Behind Being Research. We put in their announcements, they put in our announcements, and I forget the last name there. He wants to make a little announcement of what is going on there. It's very unusual. It'll take 2 or 3 minutes. Come up and make it now. Meantime, I'll find out who that April speaker is.
Hello, everyone. My name is Evgeny. I'm the president of FMBR. We are a sister organization, and thank you for allowing me to make the announcement. We are actually very happy at the foundation to have Dr. Natalia Shereko, a medical doctor from Russia, from St. Petersburg, visiting from the Institute of Biosensory Psychology. In Russia, this year the institute actually received a Guinness World Record in telekinesis, and she will be giving a weekend seminar at FMBR on January 17th and 18th. The times are 5 to 8 on Saturday and 12 to 5 on Sunday, and also the topic will be development of extraordinary abilities in a natural way, and also a seminar on Wednesday, the 21st, on self-restoration and stress resistance. And also a possibility of a seminar on theory and practice of healing work. So if anyone's interested, we're gonna be here during the break and after the meeting. Thank you.
Mike, do you remember what the other guy is? I remember who it is. Aubrey de Grey.
Yeah, that's right,
Aubrey de Grey, who was just recently on Larry King, by the way. And when he was here earlier in the year, or last year, in the fall, I had the pleasure of having lunch with him, and he's a very famous guy for good reason. He thinks that you can, with the present knowledge we have, you can find ways to engineer, repair the 7 causes of aging. There's 7 ways we fall apart when we get older, as we age. He thinks that it's one thing to try to prevent those processes, but to some extent we're going to fail. And he thinks that there exists ways to engineer, repair every one of those 7 problems. So he has something called SENS, engineered negligible senescence. Senescence being aging. Can't remember what the S stands for, but anyway, he will be here. Marvelous guy, British, Oxford, very droll, and a little bit outrageous, as the British sometimes are, and very, very brilliant. And it'll be a real special night.
When is he here? Actually, it's May. He's May, and April was the...
Saputo?
The steam block.
Okay.
The gray is May.
Yeah, I think I could talk loud enough just to remind people, if they want to see the article I gave you, I didn't have time to print out anymore, but the Genetics Policy Institute were the formulators of the first World Stem Cell Conference that was held this last year, and Bernard Siegel, who's the founder of this, will be coming to speak at our group. That's how that connection. So go to Genetics Policy Institute, and you can download the article by Aubrey de Grey, who was a guest speaker at their conference.
Right.
Okay.
Right. He spoke at Stanford, and he was great. I heard that too. I'm happy to tell you. I learned about him through John Ferber. Remember our friend John Ferber, who lives in Florida, is not often with us, but he's a very good friend of Aubrey de Grey, and they go to conferences together and hang out. Yeah, Stan. Sorry. Okay, hey, I want to bring on, we got a tremendous short report. The first event is the forum, second event is the short report, and then we take a break, and then we'll have Steve. And I want to introduce one of our most distinguished members. He really kind of stands out in a way. As a matter of fact, he's the tallest of our short reporters. I don't know if he's 6'4", 6'5", and I always know when he calls me on the phone, because the first thing he says is, "Phil, if you're having sex right now, I can call back." And I always have to disappoint him, but I'm always ready to talk to Don. The other thing about him is he's got an amazing voice. And I once kind of just kind of teasing him. I said, you should have been a singer. He said, well, I was a singer. I did musical theater. I think he told me, were you the Music Man?
No.
We're the Music Man. Okay. I thought maybe you could sing a few bars of 76 trombones. But you did sing though, right, Don?
Yeah, badly.
But you were in musical theater, weren't you?
Yes.
He was in musical theater, so it can't be that bad.
The people I performed with didn't think I was part of the operation.
Anyway, here he is, the inimitable Don Sutherland.
That's great, thank you. I'll set this down here. Thank you.
I'll take one.
All right. It's always nice. It's always nice to be talked about as whatever Phil's talking about. I notice he's talked about all the wonderful scientists and medical people that are in a group, but my name failed to be mentioned in that group of the science people, and so a lot of people have questioned my academic background and my medical background, so I'm here to cure that tonight because I don't want to take a back seat to anyone. I think at one time I told you, but I'll reiterate the story very, very briefly. When I was 18 years old, like everybody in this room, during World War II, the day after my birthday, the day after I got out of high school, I was in the service, as we all were. rallying to the colors, and they sent me to Camp Ellis, Illinois in the Medical Corps. And that's not where I wanted to be, but that's where they sent me. And so for the first they had me digging latrine trenches for the first week, and I became very proficient at that end of the business. And then they started showing us sex films. Now at that time, remember that they did not have penicillin available for anything of that sort. They didn't have, and venereal disease was a real problem. And so as a result, they showed us, I bet I sat through 200 different films on sexual diseases, and then the end of it, you know, it wasn't a pretty story. They had people that were walking around, they were in mental institutions because of it, or they were, you know, crippled and things of that sort. And so as a result of watching these things, after a while they said, we're going to send you around with a few other fellas to all of the different barracks. Now the first thing they did, they had us send around a poster like this, and I'll never forget that. The poster said, she may look like the girl next door, but. And I've never gotten, she was such a beautiful, it's the type of girl you immediately want to marry. So for a week we went around the barracks posting these things up on all of the barracks. And so that I was on my way, and I told my mother, I wrote home to my mother and I told her, Mother, I've been selected to move on up in the organization. And then a few weeks later, they said, well, now you're going to have to go out and train the people. Now today, if we turn on television, we read in the radio, I mean, the word sex is so mild today, but in the newspapers of 65 or 70 years ago, as you remember, the word sex, the word pervert, the word homosexual, the word, any of these things never even existed. And if somebody said they did, you immediately went to try to, you know, I remember reading all of, what was it, what was the great French, I read all of his books, I didn't understand them, but there was almost sex in all of them. So the first thing they did, most of the fellas and most of these other organizations were getting ready to go on leave. They'd been there for 12, 14 weeks, and the closest town was Peoria, Illinois. Now Peoria, Illinois, everybody knows how the women in Peoria, Illinois are, don't we? And so as a result, it was our responsibility to warn all of these young lads about the dangers that they were going to encounter. So as a result, along with a few other fellas, they put me on the back of the truck and they dropped us off, and it was our responsibility to explain to them the dangers that they were going to encounter from these women from Peoria, Illinois. And boy, the stories were flying wide and fast. So they gave us a broom handle, and we went around to the places and demonstrated what you did with a broom handle and a prophylactic. And it was nice because I'd never even kissed a girl. I tried to kiss a girl once, and just as I was ready, she turned her head and I got a mouthful of hair, so that's my own sexual experience. Now, the first week when I was at Fort Dix, New Jersey, we're all laying in the bunks and you couldn't get any sleep. I mean, it was impossible to get a sleep because there were fellas that were there that were 35 or 40 years old, and they had left their women or somebody else's woman at home, and they started talking about sex. Well, most of we young people thought sex was an 18-second encounter and it was over, okay? But they were talking about 30 and 40 and 50 minutes, and I said, they have to be lying. You know, that is not possible. And so I would quiz them later on, and they said that it was. So they gave us our bar handle. We went around to the various places. So they, after 2 or 3 of them, they dumped me off at one place. They dumped me off at one place, and so I went to the headquarters, little headquarters hut, and I walked in there, and there were several whacks. And that struck me as being a little, and I said, ma'am, can you tell me where your orientation room is? And so they said, well, it's building D or some building. So I went over to building D and I walked in. I froze. Here was a company of whacks, okay? And Now, I'm 18 and a quarter, all right? I'm 18 and a quarter, and all of a sudden I'm the venereal disease expert at Campbell's, Illinois. All right? The best I'd ever done was shaking hands. So basically, I'm sitting there and saying, what in God's name can I tell these people? I'm literally shocked. I mean, I'm really embarrassed. And I didn't know I was frozen. I was literally frozen. So, but I remembered all the movies, so I started throwing out words like gonococci and lymphogranuloma inguinale in the 3 stages of syphilis. And the captain there, she said, soldier, is that what you're supposed to be telling us about? Is that what you're telling the other people? And I said, no, ma'am, that's not what I'm telling. She says, incidentally, what is that broom handle like implement that I tried to tuck away? And I said, well, ma'am, I said, I pulled it out, and I said, I'm telling the other fellows this. Now, when I held that thing up, I mean, they howled, they roared, and man, they started taking me apart because they knew they had a pigeon, okay? And it was very, very embarrassing. They said, well, what do you do with that? I said, well, I'm supposed to show the fellas how to put one of these things on. And so I said, well, what you do is you insert it over the top, and this is vitally important to prevent, and I went into gonorrhea and lymphogranuloma inguinale with complications of granuloma venerea and all of those wonderful things that we all love and know. And I said, basically, this is what is supposed to be done, and we're supposed to leave a space up there for it. And they said, well, why do you leave a space up there? And what is that thing for? Some of the women said, well, you could use it for bicycle handles. You know, you could put it on a golf club. And one of the more seasoned ladies said, well, what you can do, you leave that space because when you're about to become in an amorous situation, you take some crushed ice and put it on the inside, and it keeps the swelling down. And I thought, my God, what a wonderful suggestion. Well, I'll tell you, number one, I never forgot that for the rest of my life. That's not the reason I'm here tonight. I just wanted to establish my medical credentials. I went back, and everybody in my company wanted to know how things were. And the colonel, who was an eye surgeon from Chicago, came in and said, soldier, how did it go? And I briefly told him, I said, you know, and this colonel, my God, that was like talking to God. And I briefly explained it to him, and I said, you know, Colonel, I would like to request a transfer to the infantry. And within 3 days, I was on my way to the transfer to the infantry, where I really held the fellas back up there. My point of being here tonight, incidentally, is not that. Pam, who used to sit over there, who comes in from up north somewhere up with you people, was telling me a lot about a little website called goodcausewellness.com. And on goodcausewellness.com, I went on to buy, is it Selva seeds, the seeds that come from the Mayans?
S-A-L-B-A.
What is it?
S-A-L-B-A.
S-A-L-B-A. So on there, I saw something and it says, the Health Ranger asks questions. So I hit the Health Ranger, ask questions. It took me to another page. And on it is the Health Ranger's interview with about 67 different people, and each one of them takes about 12 to 15 minutes. Well, the first one I hit was Dr. Stephen Sinatra, an old friend of yours, Mike. Now, Stephen Sinatra is probably one of the foremost cardiologists in the United States, has written perhaps a dozen different books on cardiology. And and I would advise going on and listen to it. It'll only take about 12 to 15 minutes. So everybody that comes into my office, and my office is not a health office, and matter of fact, most of the people come in and don't even want to come in, but I drag them in, I make them listen to this. I make everyone that walks into my office, I make them listen to that, because he is the author of a book called The Sinatra Solution, Metabolic Cardiology, and it should be listened to and should be read, because we all know that all of us around are going to die of some aspect of that normally. And what he talks about, rather interestingly, and he does it in a very nice way, is that Mike, he's been up to the meetings up in Las Vegas, those 3 or 4 day meetings, almost every year, if I'm not mistaken. He was there this year, okay. Well, his book is well worth reading and well worth getting and reading, but in it he talks about the 4 things that are important to pump ATP, or literally oxygen, into the heart and into the body. And those 4 things are, number 1, magnesium, because as we know, for 1 million years, man had a magnesium intake into the body of about 2.5 times calcium. And the calcium, if we've ever seen a fire hose, calcium constricts, magnesium relaxes. Calcium constricts, magnesium relaxes. So that's what he says is number 1. Number 2, he recommends L-carnitine, D-ribose, and coenzyme Q10. The heart muscle is 70%. The heart muscle is 70% coenzyme Q10. If we take statin drugs, it destroys the coenzyme Q10 and helps to destroy heart muscles.